So today's focus is going to be on the structure of proteins, the building blocks of proteins. If you remember, everything's made of monomers. The monomers of proteins are called amino acids. And amino acids have the following structure. You have a carbon in the middle. This is called, and you might want to add this to your notes because I don't know that there's anywhere to write it. It's called the alpha carbon. That's the Greek symbol for alpha, little fish there. Connected to one side of the alpha carbon is an amino group. This is where the amino comes from in amino acids. On the other side of the alpha carbon is a carboxyl, or remember this is also called a carboxylic acid group. So this is where the acid part of amino acid comes from. So they're called amino acids because they all have an amino group on one side and a carboxyl group on the other side. A hydrogen connected to that alpha carbon. And in the last spot is an R group. Remember, we use the letter R to represent a variable that multiple things can go there. In the case of amino acids, there are 20 different things that can go there. So there are 20 different R groups. On the next slide, I'm going to show you some examples so you can see what I mean. But the different R groups give the amino acids their specific names. Like you have lysine and glutamine and arginine, methionine. The names of the amino acids come from the R group that's on them. So I'm going to give you a moment to sketch this and get this information, and then we'll move on from there. All right, so this diagram is showing some of the amino acids with the R groups there. So for example, I'll highlight them. So this could be an R group. This is called valine. The amino acid with this R group is called leucine. This one's called isoleucine. This one's called phenylalanine. This one has this long R group is called lysine. So, no. What you need to understand is that there are 20 different things that can go in that spot. You do not have to know the 20 things. That's what makes each amino acid different. In every other way, every amino acid is the same. A protein chain of amino acids can be anywhere from, some of them are 300 to 1,000 amino acids long. So depending on how many amino acids are in the chain and the order of the amino acids in the chain, that's going to determine how that protein ultimately folds into a shape and what its job is going to be. Notice here, it says that these amino acids have hydrophilic R groups. The one above it has hydrophobic R groups. So some R groups are going to repel each other, some are going to be attracted to each other, and this is going to determine how that particular protein is going to fold up and what its job is going to be. So this is just an example, but no, you would never be asked which one of these is valine. That would not be a question you would ever be asked. But this is just showing a sample of some of the different R groups that can be there. All right, so moving on. What hooks the proteins together into a chain, they're called peptide bonds. And a peptide bond is just the same exact reaction we've been talking about. Condensation or dehydration synthesis reaction. OH and H, hydroxyl and hydrogen. The OH from one reacts with the hydrogen from the other. They form a drop of water. That water is going to leave. And what you're left with is now going to be a peptide bond, 2 amino acids connected together. Eventually, in a protein, you would end up with 300, 500, 1,000 amino acids connected together, all hooked together by the same bond. And remember from yesterday, what tells your cell what proteins to make? Your DNA. So your DNA is going to say, add a lysine here, now add a valine next, now add a leucine next, now add a methionine next. And it's going to determine how many amino acids, what order of amino acids, and therefore what proteins your body's going to make. So what is the function of proteins in your body? We remember lipids were for energy storage in general, and then the steroids. Carbohydrates were for quick energy. Proteins form most of your body's structures and carry out a lot of your body's functions. Every enzyme in your body is a protein. That's what speeds up reactions. So all the enzymes that are involved in digestion, that are involved in cell functions, that are involved in transmitting impulses in your brain. Other than steroids, all the hormones in your body are proteins. So insulin and growth hormone and calcitonin and all of these other hormones that are proteins. And then things like keratin, elastin. Keratin's what your fingernails and your hair are made of. Elastin's what makes your skin elastic. Another one you've probably heard of is collagen. That's a protein. Hemoglobin, that carries oxygen in your blood, is a protein. Pigments like melanin that give your skin color are proteins. Antibodies that fight off diseases are also proteins. Iron's in the middle, and then it's a protein structure. It's called a heme group, and it's a big protein with iron in the center. Yeah, so iron is in the middle of a hemoglobin. This is a picture of insulin. Look how big and bulky, and it looks like a blob. But the truth of the matter is, it starts off as a chain, the same chain of amino acids that I just showed you, but then it folds up into a specific shape because of those R groups that I was talking about. They're going to determine how it folds up. So even though in this picture, this looks like it's just some kind of a blob, it's actually an intricately folded shape that is just like a key fitting my front door. Insulin specifically attaches to a receptor on cells and causes your cells to then take in sugar. And every hormone and enzyme in your body works the same way. On first glance, like I said, it may just look like a blob, but it's not. It's actually got a very specific structure. So these are some examples of what proteins do in your body. So the goal of our activity today is that we're going to simulate how proteins go from starting off as just a chain of amino acids to this complex folded shape. That's what we're actually going to be simulating in the lab. So all proteins start off with, these are actually just some other examples. So enzymes that speed up reactions, they can store things. Antibodies that fight off diseases are proteins. In your cell membranes, you have transport proteins. Hormones, muscles are made of protein. Receptors, like that pick up nerve impulse information. And structural proteins, like keratin, collagen, things like that. So proteins can be described in levels of structure. And this is actually the last one we're going to do before we move into our lab groups. The first level of structure is called the primary level. And the primary structure of a protein is the order and number, technically, of amino acids that there are in the chain. You can recognize, you could actually look at a protein chain if you were just looking at the primary structure, and you could count how many amino acids there were by looking for this NCC. Because it's going to be the amino group, the alpha protein, the carboxyl group. Amino group, alpha protein, carboxyl group. Amino, alpha, carboxyl, there's a third one. Amino, alpha, carboxyl, that's a fourth one. In this one, they don't have the letter R. They actually have the R groups there. So these are actual amino acids. So this is an R group. This is an R group. This is an R group. That's an R group. So each one has these R groups attached to it. They're just not showing it as the letter R for this particular thing. Okay, so once the primary structure of a protein forms, once the protein is built, which is what you just did, the next thing that happens is that along the chain, some of the amino groups from one amino acid react with the carboxyl groups from another amino acid, and they form hydrogen bonds. When they do this, you end up with repeating patterns along the protein chain. There are two main kinds of patterns. One is called an alpha helix. It actually looks kind of like a curlicue. It's also written like this, alpha helix, that same Greek symbol, alpha helix. Again, what you see here is these little dots. These are hydrogen bonds forming. Now, this doesn't happen along the entire chain because, remember, that also connected to these carbons are some kind of R group. And some of these R groups are big and bulky and get in the way. So in a real protein, you would see along this chain of 300 amino acids, little sections of that protein where you might see a pleated sheet, which looks like that, or an alpha helix that looks like that. So to simulate that in the lab, what you're going to do is take your pipe cleaner, wrap a section of it, any section, around your finger and create an alpha helix. Again, in a real protein, you might find several of these in different locations along the chain. Somewhere else on your make a fan out of paper. You kind of go back and forth in a zigzag, and then pull it apart. And that would represent a pleated sheet. Again, in a real protein, there would be multiple areas where you'd have these little repeating patterns, specifically because of hydrogen bonding between the amino groups of some amino acids in the chain and carboxyl groups of amino acids further down the chain. So go ahead and do that step now. So the next thing that happens in a protein, after these little repeating areas form, in fact, if you look at this picture, you can see this area? This is an alpha helix. And see this little area here? These are pleated sheets. So you can see in a final protein these patterns in sections. But the next thing that's going to happen is that the entire chain is going to fold up. That's called the tertiary structure. What causes the tertiary structure is the R groups now interacting with each other. So remember how every amino acid has an R group. Those R groups, some are hydrophilic, some are hydrophobic, and they're going to basically react with each other and cause the entire chain to fold into a final shape. One of the things that forms, that stabilizes proteins, are what are called disulfide bridges. Those come from the functional group we talked about called a thiol or a sulfhydryl group. If you remember sulfhydryl or thiol, it's SH. It forms, here's a picture in one. It forms a covalent bond and forms what's called a disulfide bridge. So the entire protein folds up into a final shape, a specific final shape that will determine the job of that protein. I added a little note here that if you change the pH or you change the temperature, especially if you heat it up, like when somebody gets a really high fever, 106, 108, these are all very delicate. This folding, these hydrogen bonds, and these interactions. What will happen is the tertiary structure of a protein can start to unfold. So if these are proteins that are necessary for nerve impulses in your brain, and they unfold so they can no longer do the job they're supposed to do, it can basically kill you, or it can cause brain damage. So tertiary structures are delicate. This is why we need buffers to keep our pH stable. This is why we can't have really extreme high temperatures, because it will cause these structures to unfold, and if this unfolds, it won't have the proper shape anymore. So every protein has a primary structure, a secondary structure, and a tertiary structure. The primary structure is the order of amino acids. The secondary structure are these little repeating patterns that I showed you here. There's one, here's some. And then the tertiary structure is how the whole thing folds up. Some proteins stop there. The final structure of the protein, it's done and it's ready to do its job. But there are also some proteins in your body that have one more level of structure. And those proteins have what's called quaternary structure. Quaternary structure is where the final protein consists of multiple chains. For example, hemoglobin, which you see here in this picture, actually consists of 4 separate chains. They all hook together. Iron sits in the middle of it, and you end up with a functional hemoglobin molecule. Insulin actually is stored as a protein consisting of 6 parts. So what you're going to do to simulate this is you're going to hook everybody in your group, you're going to hook your proteins together and simulate a quaternary structure for your protein. Again, not every protein has one. Some proteins, their final level is tertiary. They're made of just one chain. But there are several proteins that are made of 2, 3, 4, or 6 chains all hooked together. So that's quaternary. I see your hand. One sec.